We're going to look at 5.45, and also we're going to talk about how to make a cladogram. But let's just briefly talk first about how we can look at specific molecules and create a cladogram to show the evolutionary relationships of these different taxonomical groups. So the example that they use quite often is cytochrome C as a way to divide organisms. Cytochrome C is a protein that you find on the mitochondria, and it's an electron carrier in the cell respiration process. Another very popular protein that they look at, especially when we're looking at mammals, mammals I would say, is hemoglobin, the protein that helps us carry oxygen around. I should say probably not limited to just mammals. But anyway, so because DNA mutates, we can determine how much the DNA has mutated by how the protein has changed over time, right? Because DNA, how many times have I said this? DNA equals protein. So if the DNA changes, then we should see changes in the amino acid sequence of the protein. And the more changes there are, the more differences we should see in these species. So this is a group of primates. These are relatives of humans, also sometimes monkeys, but it also includes organisms like lemurs. But these are all different types of primates. So if we are looking at this, we can conclude, or at least predict, that this organism here, or it could be the lemur, would have the most differences than these different primates down here. Here we have Homo sapiens, which is us, Pan troglodytes, which is chimpanzees, and then we have Pamponiscus, which is bonobos. Those are very similar. All three of these are very similar in DNA structure. But as we work our way down this way, we'll see more differences. So we put them in different groups. Second, we can also identify how long ago the changes occurred. So the more changes in the amino acids of the protein, the more mutations that have happened in the DNA, and we can predict using like molecular clocks how long ago those divisions or those separations into different species took place. When we talk about species dividing, we call this speciation. So here we have an example of speciation. Here we have a time when we had speciation. Here we had a time we had speciation at these different nodes. So that's how we can use variations in specific molecules, specifically cytochrome and also hemoglobin, to determine some sort of evolutionary relationships. Let's move on to making a cladogram. Okay, let's talk about a cladogram first. So you can do this two ways. Both of these models are the exact same thing. We have species A, B, and C. Here we have B, A, and C. They're just in different order. We have a node down here that's a common ancestor between C, B, and A. And we have a node down here that is a common ancestor between C, B, and A. We have a node here that's a common ancestor between A and B. And we also have a node here that's a common ancestor between A and B. So they're exactly the same. And now, okay, so now we're going to talk a little bit about how to make one of these based on information that they give you. So let's go to a slide. This is a typical table they'll give you and ask you to construct your cladogram. So the way I do it is I pick the organism that has the least amount of characteristics. All right. So we have 4 different organisms, right? We have a shark, we have a frog, we have a kangaroo, and we have a human. So they did this sequentially. It's not always this easy where we can see, 1, 2 different characteristics, 3 different characteristics, and 4. Sometimes it's a little bit more scattered, and I promise I will give you some practice. And I'm going to do the model where it's kind of like a branching effect, where we just have a line and the organisms come off each end. So at the beginning here, we have a common ancestor between all of them. So as evolution is occurring, we had speciation occur at this place. And when speciation occurred, the sharks, just going to use the letter S, branched off into their own group. And as this one developed along this way, it developed different characteristics, different physical characteristics. So the shark went off this way, but there were some organisms that developed lungs. So here we have another node, and there was a group of organisms, amphibians such as frogs, that developed lungs. So frogs have both vertebrae and lungs, where sharks only have vertebrae. And then evolution occurs. Then we get into our mammals, and we have a group of organisms that are able to produce milk in mammary glands. Some of those became kangaroos. These are our marsupials. And some of them evolved further to carry their babies inside for the whole time. The difference between a kangaroo and a mammal that we're familiar about is a kangaroo, before the baby is completely finished in the oven, it spends some time in the mother's pouch, where placental mammals, the majority of the mammals on this planet, carry their offspring to full term. So we'll put humans there. So sharks have the least amount of characteristics, and as you work your way up this cladogram, you get more and more characteristics as they branch off. So that's how a cladogram works.